Have you ever wondered how scientists can peer into the living brain, watching thoughts and actions unfold without ever making a single incision? It is an incredible feat of modern neuroscience, allowing us to map activity with remarkable precision. At its core, functional magnetic resonance imaging, or fMRI, is enabled by the precise alignment of hydrogen protons within the brain. This alignment allows the scanner to detect changes in blood flow, which are directly linked to neuronal activity. The process begins because our brains are rich in water, and water molecules contain hydrogen atoms, each with a single proton that acts like a tiny magnet with a spin. When a person enters the fMRI scanner's powerful magnetic field, these randomly oriented protons align themselves either parallel or anti-parallel to the main magnetic field. A radio frequency pulse is then briefly emitted, knocking a small fraction of these aligned protons out of their alignment. Once the pulse is switched off, these protons relax and snap back into alignment with the main magnetic field, releasing energy as they do so. Crucially, the rate at which they realign and releases this energy, known as their relaxation time, is sensitive to their local magnetic environment. This local environment is subtly influenced by the presence of oxygenated versus deoxygenated blood. So, how does this translate into seeing brain activity? When a brain region becomes active, it demands more oxygenated blood. This influx of oxygen-rich blood actually overcompensates for the oxygen used, leading to a temporary local increase in the concentration of oxygenated hemoglobin. Oxygenated hemoglobin is less magnetic than deoxygenated hemoglobin, which subtly alters the local magnetic field around the hydrogen protons. This change in the magnetic field affects the protons' relaxation times, specifically the T to star relaxation time, causing them to realign slightly slower and emit a stronger signal. The fMRI scanner detects these tiny differences in signal strength, allowing researchers to create detailed maps of brain activity. This phenomenon is precisely what we refer to as the bold signal, which stands for blood oxygenation level dependent. It is a remarkable indirect measure, showing us not the neurons firing directly, but the metabolic consequences of their activity. The T to star relaxation time is particularly sensitive to these local magnetic field variations caused by the differing magnetic properties of oxygenated and deoxygenated blood. Understanding this intricate interplay between water protons and blood oxygenation provides the foundational insight into how fMRI can visualize brain function. Ultimately, the subtle dance of hydrogen protons aligning and realigning in response to magnetic fields and blood oxygenation provides a non-invasive window into the brain's complex workings. This allows us to connect specific thoughts and behaviors to their underlying neural substrates, advancing our understanding of both healthy brain function and neurological disorders.